do a full launch today. Photosynthesis and respiration, as I've told you, I think are the two most complicated units that we're going to hit all year. So just be aware, it's going to be a grind. I will tell you how I approach it, and what I hope is that you can find this kind of fun, because I think it's really fun. It's a different kind of challenge from stuff that we've done before, but it's going to be something that many of you are good at, and it's a skill that I think a lot of you bring in here from your other classes and your outside life. So I don't think it's going to be super challenging for you. It's probably harder for me than it is for you, because it's not something that comes naturally to me. So we'll talk about that when we get there. With your Chromebooks, if you haven't already turned in the yeast catalase activity, will you do that? And what I want in particular is a picture of whatever you've got. If you haven't turned anything in on Canvas, I went through and I erased all the zeros, and I want you to take a picture. If you still need more time, you can have more time. I think the work that we did on the yeast catalase lab and the work that we started on the photosynthesis and the ATP POGL, I'm gonna just not worry about due dates for any of that stuff, because it's a lot of work, and I know that you all have a lot going on. What I want is for us to all get kind of on the same page. I'm behind right now with my grading, so I have a lot of sympathy for those of you who are feeling overwhelmed by school right now. So don't worry about late penalties, but I want those pictures. So turn in what you've got, even if you haven't turned in anything else. If you haven't turned in anything, you can take a picture of whatever you have for the yeast catalase after. And then I made a major change that some of you might need several weeks to adjust to. I moved the turn-in bin over here, and the stapler and the hole punch and stuff are over here now as well. And the reason for that is because it's hard to get to up here, and I think because it was hard to get to, people kept grabbing the stapler and taking it, so then I couldn't find one. Anyway, I'm hoping that this will work better. So when you go to turn in your stuff, you can either pass everything to the west end of the room and have somebody there turn it in, or you can quietly, quickly get up and turn things in, or you can just wait until it's a good time. But that will be our new turning in procedure. If you need some sort of processing time or counseling to deal with the relocation of the turn-in bin, let me know. I will schedule you. We could do a group counseling, group therapy. All right, any questions about any of that? Okay, I'm gonna start. If you are taking pictures, that's great, do it, get them turned in, but then once those pictures are done, close your Chromebook. You're gonna need something to write with and write on, but I'm not gonna have you take a ton of notes today. Instead, what I'm gonna do is show you a couple of things that need to be in your notes, and then you will have a bunch of time today to work. And as part of that work, you can copy the relevant pieces of information into your notes, or you could do it outside of class, whatever works best for you. I'm gonna go, this is the official Brigar presentation of photosynthesis information. Oh wait, hang on, there's something I wanted to show you first. As we start going through this, remember that course and exam description is really the go-to for determining what's going to be on the test. Now, what I've told you is anything that I say could be on the test is fair game. I may ask you to learn a little bit more sometimes, or a little bit less sometimes. But if you're ever wondering, wait, what is this going to look like on the exam? Remember, the course and exam description is there. It's linked in the modules, and you can just look it up too. So what we've done so far has been pretty simple. So if we start with unit or section 3.1, you can see it just talks about structure and function of enzymes, and and the substrate and the active site. You already knew that stuff. We went through that quickly. Environmental impacts, this had to do with denaturing proteins due to temperature or pH. And then we talked about relative concentrations of substrates and products and enzymes. That's what you just did with the yeast catalase lab. And then on Wednesday, we went very quickly over inhibition, competitive and non-competitive inhibition. You can see that's all there. When we get to photosynthesis, here's the stuff you need to know. All of this, energy related, no, sorry, this is energetics. This is cellular energy. So this was the stuff that we went over about the laws of thermodynamics and the conservation of these core metabolic pathways. And then when we get to photosynthesis, it gets crazy. Like this is what you know already. Photosynthesis uses carbon dioxide and water to make carbohydrates and oxygen as a waste product. And this is all the stuff you need to know. You need to know the structure of the chloroplasts. You need to know where the light reactions happen, where the Calvin cycle happens. You need to understand photosystem 1 and photosystem 2. You need to understand the electron transport chain. We're learning about a new molecule, NADP/pH. And then we're going to look at ATP synthase, and then how the energy from the light reactions is stored and used in the Calvin cycle, which doesn't produce energy, but it moves that stored energy into carbon compounds that ultimately will become sugar. So that is crazy, and that's what we're going to do today. I'm going to do my part quickly because my part shouldn't really be that critical for you. What I hope to do is lay out what your work should look like, and then ideally that will end up allowing you to target your work during class and outside of class so that you're prepared for both my test and the AP exam. All right. Any questions about any of that before we go? Okay. It starts with this. This is the stuff you already know. Photosynthesis is a process. It happens in the chloroplasts of any organism that can photosynthesize. There's also bacteria that can photosynthesize, and those don't have chloroplasts because they don't have organelles, but they have similar, maybe different systems, but the basic idea is the same. Our general balanced equation for photosynthesis is you need carbon dioxide and water and energy from light, and all of that is used to make glucose and oxygen, which is a waste product. We use oxygen, but the original photosynthetic organisms were just producing it as a waste product. We'll look later at how that actually changed the entire planet. Okay, so this you should already know, and if you don't, make sure that you have this written down. It uses energy from sunlight, and that energy gets stored in the molecule glucose. This is what you should have known prior to coming into this class. Now what we're going to do is we're going to look at how this happens. And we're not going to dive into as much detail as we could, but it's going to be enough, okay? We've talked a little bit about this whole solar energy, and this involves something called a pigment, and most of you probably know this already, but pigments are things that absorb light. Pigments absorb some light and reflect the rest of it. So for example, if you have a pigment that absorbs greenish to sort of dark bluish light, then what it's going to reflect back is yellowish and reddish light. If you have a pigment that absorbs reddish light, then what you will get is something that reflects back yellowish and green. So when we look at the absorption spectrum, this is a graph showing which frequencies of light are absorbed. If we look at chlorophyll, which is green, it absorbs blue light and red light. And that's why plant leaves largely look green. They're reflecting green. Plants look green because that's the thing they don't absorb. There are other types of chlorophyll that absorb a little bit more green light and lower frequencies of red light. Those are present in most photosynthetic organisms to a certain degree, so you get this kind of combo. And then there's also other pigments. They're not chlorophylls at all. There's xanthophyll, there's beta carotene. Those absorb completely different spectra. So like beta carotene absorbs mostly green light, and so it ends up looking reddish or purple or yellow. And so when you look at plant leaves, oftentimes you can see plants that are multicolored, and what we're seeing here are all the various pigments. Many of them are still important for photosynthesis, okay? All right, here's a diagram. You're probably not going to be able to get everything down here, but you're going to need to know virtually all of this. So this is a diagram of a chloroplast. I'm going to go through it pretty quickly, and then if you need more time to complete this, you can come back and look at the presentation and get this written down in your notes separately. So in the chloroplast itself is one of those membrane-bound organelles. It has two membranes. There's an outer membrane and an inner membrane. And there's a little stuff in between it that isn't really important for our purposes. All the good stuff happens inside the inner membrane of a chloroplast. Within a chloroplast, there are more membrane-bound structures. So this is like a third layer of membranes in a way. And each one of these little flattened membrane-bound discs is called a thylakoid. If you take those thylakoids and stack them up, then all those thylakoids together are called a grana. And that, I don't know if that's something that you're going to need to know on the exam. That isn't something that's particularly important to me. Sorry, this is scratching the back of my neck. That's not something that's particularly important to me, but I want you to at least, if you see that term, understand what it is. And these are terrible words, because even this, again, what I mentioned to you earlier, is this is something that doesn't come naturally to me. Memorizing this kind of stuff isn't easy for me. Like, to me, I look at thylakoid and I look at grana, and I don't look at those words and think, oh yeah, thylakoid is the singular and grana is the plural. Like, that doesn't jump out to me as something obvious. They weren't named the right way because people didn't know what they were looking at the time. Having said all that, like, it's memorizable, and you can memorize it at least short term now and maybe short term again before the exam. Inside the thylakoid membrane is a fluid filled, like it's full of fluid, and that area inside the thylakoid is called the lumen. All right. Outside of those thylakoids is more fluid filled stuff. That part of the chloroplast is called the stroma. So far we have outer membrane, inner membrane, stroma, and then we have the thylakoids, which form stacks called grana, and inside the thylakoid is something called lumen. So yeah, it's a lot, but you'll need to know all of this. You'll need to know the anatomy, so to speak, of a chloroplast. We'll talk more about what happens inside here in just a second, but the basic idea is the light reactions, which is where the solar energy gets into plants and is initially stored in chemical compounds, happens in the thylakoids. And then in the stroma, there's a bunch of chemical reactions that are collectively known as the Calvin cycle, and that's where the glucose is made. They used to call this the dark reactions, but as in like this happens when it's light, and this doesn't need light, but then people thought this only happens during the day and this only happens at night, so now they usually don't call them the dark reactions anymore. Yeah.
And by sugar, it's a 3-carbon sugar?
Yeah, so we'll talk about that. It's a little bit complicated, but that's one thing that they have mercifully allowed us to just not worry about. So this is the chloroplast overview, and this is kind of the big picture of photosynthesis. You can see our inputs here of light and water, and you can see our outputs of, light, water, and carbon dioxide, and we can see our outputs of oxygen as waste and sugar, which is the main product of photosynthesis. Yeah.
Did you say lumens are a bunch of molecules inside?
Lumens is the inside part of the thylakoids. It's like the inside of a cell or an organelle. This isn't technically an organelle. It's a membrane-bound structure within an organelle, but it is filled with all sorts of good stuff, enzymes and proteins and sugars and all the stuff that you find in a cell that are also located inside these thylakoids. Not DNA, but they have. The lumen is basically the cytoplasm of the thylakoid, and the stroma is the cytoplasm of the chloroplast. Okay, other questions? All right. So the next thing we're going to look at is, this is just an overview of the thylakoid membrane, and you don't need to write anything down here. This is a simplified version of what we're going to focus on first, which is the light reactions. The light reactions happen in the thylakoid membrane, so there's some stuff here that's green. Those things have chlorophyll in them. That's where the light gets captured. And then there's this electron that it's showing kind of moving around through all these blobs. Those electrons carry the energy. The energy from the light is absorbed by the pigment, and that energy is then transferred to an electron where it becomes more energetic. Okay. Here is what you will need to have written down, but you won't have time to do it right now. So this is essentially the sum total of what you're going to need to know about the light reactions, and I'll show you a little bit more details and explain a couple of the things that happen, but this diagram you're going to need to know, all right? So first, remember that the light-dependent reactions happen in the thylakoid membrane. So if this is a chloroplast and this is a thylakoid, these things are embedded in the membrane of the thylakoid. This side on this diagram is showing the lumen of the thylakoid. So this is the inside of the thylakoid, and this side is the stroma. This is the inside of the chloroplast, but outside of the thylakoid. Okay? And the whole process, I'm just going to go through this and just tell you, this is the stuff you're going to need to know, all of it. Okay? So the first thing is, we tend to think of this as sort of being a sequential thing, but the reality is that all these things are happening at the same time. And there's a bunch of different kind of ingredients and products that happen. One critical piece is we need to have an electron. The electron is going to be what holds on to the energy, and then it's used, that energy is used to do all sorts of stuff. That electron comes from water. And you don't need to know the details of this, but what happens is the water is split enzymatically. That enzyme splits that water to produce some oxygen, some protons, hydrogen ions, and an electron. You don't need to understand the details of that reaction, nor do you need to be able to pencil it out. It's just the input is water, the output is oxygen, hydrogen ions, and an electron. We'll come back to these momentarily. The electron ends up going into an enzyme, a protein called photosystem 2. This you'll need to know. So it's called photosystem 2. And it goes in there before it goes into photosystem 1, and that's because these weren't named by function, they were named by when they were discovered. Probably we saw photosystem 1 first, and then we saw photosystem 2, and we didn't know what they did. And so the long and short of it is, the process starts in photosystem 2. And what happens with that electron is inside this enzyme, that there is a pigment, and this P680 identifies the pigment. You don't need to know that. that photon, which is what light is, it's just a bunch of particles called photons, is captured by the pigment and used to increase the energy of the electron. So you can even see here visually that this electron starts down here and moves up here. I don't know where the electron is physically in this molecule. We're looking at it energetically. So the electron has lower energy here and then zip, higher energy here after it is powered up by the photon. It then moves through this purple stuff. And the purple stuff has a name. There's several names because there's several pieces to it. In general, we call it cytochrome or the cytochrome complex. You don't need to know that. You'll see it in just a second, but that's not part of the stuff that you need to know. You do need to know that as this electron moves through these things, it's literally getting passed from one molecule to the next, to the next, to the next. And as that happens, it becomes less and less and less energy. The energy that was there is used to pull more protons into the lumen. So now we have protons from splitting water. We have protons that are brought in through this section of the electrons travel. Yeah?
So is the methane gradient in the hydrogen ions meant to be an enzyme?
It is an enzyme.
And do the electrons essentially work like ATP would for numbers?
Sort of, like, we don't need to know the details, and it's really good for all of us because I don't know the details. I don't know exactly how that chemistry and physics works. Yeah. So you certainly don't need to know. If you want to, we could look into it. I'd love to look into it more, but I don't know off the top of my head exactly how the reactions work. Okay, and then that electron eventually is transferred to another enzyme, which you'll need to know. It's called photosystem 1. And in photosystem 1, which has a slightly different pigment in it, it gets struck by another photon, which bumps up the energy level again. So there's two charges. One charge in photosystem 2, the energy from that charge is used to pull hydrogen ions in, and then one charge from photosystem 1, and that energy gets passed through a couple of other enzymes that we don't need to know the name of, and it's used to reduce something called NADP plus to something called NADPH, and we'll come back to it. This is the stopping point for the electron. This whole path that the electron takes is called an electron transport chain. And sometimes we just abbreviate it, oops, not electronic, electron transport chain. And sometimes we just abbreviate that ETC. And there are other electron transport chains in living organisms. This isn't the only one. This is the electron transport chain in photosynthesis. Next week, we're going to look at one that's in respiration. So if somebody just says, like, tell me about the electron transport chain, the correct response is which one, because there's many of them. But in this one, this is the path that it follows, and you'll need to know all of that right down to here. This is where the electron ends up when it's all said and done in this molecule called NADPH, which we'll come back to, all right? And then in the meantime, we've had some hydrogen ions produced from the splitting of water, and we've had some hydrogen ions brought in by this cytochrome complex, and that ends up creating a concentration gradient. There's more protons, there's more hydrogen ions in the lumen, on the inside of the thylakoids, than in the stroma, outside the thylakoids. That concentration gradient is used much the same way that, like, secondary active transport is used. In this case, what happens is those hydrogen ions flow with their concentration gradient from high concentration of hydrogen to lower concentration of hydrogen. And as they do that, they literally make a molecule, an enzyme called ATP synthase. They make that spin around. And as it spins around, it attaches inorganic phosphate molecules to ADP to create ATP. So we get two things from the light reactions that are really important. One is NADPH, which we'll come back to. One is ATP, which you already know about, but we'll come back to ATP synthase at least briefly. The other thing that we end up with is oxygen. This is a waste product. Oxygen gas can diffuse right through cell membranes and plasma membranes in general. So what happens is this oxygen just leaves and eventually leaves the whole organism. So that's why photosynthesis produces oxygen. These plants in here right now are doing this, and as part of it, they're just busily manufacturing oxygen molecules that then diffuse right through all the membranes, cell walls, everything, and are just like come into the air. We are breathing molecules of oxygen that have been produced by those two plants right there. Okay? So if you got this, fantastic. If you didn't get all this written down, come back, because you'll need it. All right? Here is a slightly more complicated diagram. I'm not going to make a big deal out of this, but I'm just going to show you where a couple little pieces that didn't show up in that last one. So here's where we identify what's going on here as this is cytochrome. These are related molecules, and that's why this whole thing is sometimes called the cytochrome complex. We see here that this little set of molecules that makes the NADPH, it has its own name. There's an enzyme here called NADP plus reductase. You don't need to know that stuff. But for those of you who like a little bit more of the details, there it is. Okay, I want to briefly talk about this piece, the production of this molecule called NADPH. And you don't need to really understand the details of the chemistry. You don't need to know the chemical structure of this. This is what it looks like. This is actually, there's a related thing called NAD plus and NADH that we'll look at in respiration. But this is what NADP plus looks like. This is what NADPH looks like. And what you can see is that to create NADPH from NADP plus, we need to add some electrons. It actually hooks hydrogen on as well. So we add a hydrogen, but we also add 2 electrons, and it becomes electrically neutral, where NADP plus is positively charged. Those electrons have energy in them. This is a reduction reaction, so you might remember that from chemistry. Reduction just means that the product is more negative than the reactant. So this is the reactant, this is the product, this has 2 more electrons in it than the reactant did. I think it's silly that they make such a big deal out of this being a reduction reaction, because that doesn't probably mean a whole ton to you. Again, I'm not a chemist, so it doesn't really mean that much to me. But whenever biologists emphasize that something is a reduction reaction, what they really mean is we're sticking electrons to it, and electrons carry energy. So what we're really doing when we have a reduction reaction is storing energy. And that's what NADPH is. NADPH is a molecule that stores energy. It's a lot like ATP. It's not as useful as ATP. It is used in a couple specific places in cells. First, ATP is used all over the place. But it is similar, it's analogous to ATP in that when you stick those electrons and hydrogen on, the resulting molecule has more energy, and it's pretty reactive, so we're going to use that energy, and we'll see how momentarily. Okay. And then the other piece that I want to talk about with regards to the light reactions is this ATP synthase. So I already went through the first part of this, so hopefully you understand, like, what I hope is that you can visualize the diagram that I just showed you. But basically what happens is, because you're splitting water and pumping protons in to the lumen, we get this accumulation of hydrogen ions, and then that hydrogen then diffuses along its concentration gradient through an enzyme called ATP synthase. And as that happens, the ATP synthase uses the energy that was stored in that concentration gradient to put together ADP and an inorganic phosphate. So it uses the concentration gradient energy there to make ATP. And we'll see in a few minutes. What's going on here is actually a mechanical process, which is really neat. There's a video that's posted here, but I'm not going to play it yet. I'll play it when we're done. But you can see that, like, there's this little arrow here in this image. It actually, like, spins around like a little ratchet. It's kind of crazy. All right, so light-dependent reactions, check. I expect at this point that you, I hope, understood what I said and probably can't remember any of it. Or at least some of you can't remember any of it. Here's your goal. Your goal is to be able to reproduce that diagram that I spent the most time on and explain what happens. That you'll have to be able to do. Photosystem 2, photosystem 1, water splitting, production of an electron, photons, jacking up the energy of that electron, protons in the lumen, more protons coming into the lumen. The whole thing, you should be able to draw that whole thing and explain it all. That's your goal. Yeah.
So is the ATP synthase meant to be spinning fast enough so that the ADP and phosphate group combine?
It's not so much that, it's just that it's energy transformation, so that it starts with energy stored in a concentration gradient. That energy gets used to make this thing physically move. That's a form of energy, and that physical motion is then used to attach these two things. Again, I can't tell you exactly how it works because I just don't know, and you certainly don't need to know exactly how it works. It's just, to me, it's kind of a cool thing. Like, I just sort of visualize it as like a pump almost, like the like water going through something and making something else spin, and then that something else spin is doing something interesting, and that's really what's happening. You'll see the video, but the video won't go into the details of the reactions either. Okay, so this is just back to a quick overview, because what I want to highlight is that we've looked at two of our inputs so far. We've looked at our input of water and our input of solar energy into the light-dependent reactions that take place in the membrane of the thylakoids. And now what we have in terms of outputs is our main waste product of oxygen. And we've made some ATP and NADPH. These have energy in them. This is waste, these have energy, but we don't have anything that is super useful to other organisms yet. And it's not necessarily all that useful to the photosynthesizing organism either. ATP is great, but ATP that's inside the stroma of a chloroplast isn't all that helpful. So what we need to do, what the plant is ultimately going to do, is take the ATP and the NADPH that was created and use that to make sugar. Sugar is relatively stable and easily transported. So it's a super useful form of energy. It's not as reactive as ATP, but that's good if you're not looking for something to react. It's kind of like the difference between a lit match, and a battery, right? Like, a battery is great for energy storage, but it doesn't do anything right away. A match is really good for doing stuff, but it's not easy to transport. ATP and NADPH are like a match. Glucose is like a battery. All right, the next thing that we're going to look at is this part of photosynthesis, which is a whole series of reactions called the Calvin cycle. And that happens in the stroma of the thylakoid, or of the chloroplast, outside of the thylakoids. The light reactions are happening in the membrane of the thylakoids and involve the lumen, the inside of the thylakoid, and the stroma, the outside of the thylakoid. The NAD and NADPH and the ATP that's produced are dumped into the stroma where they can be used in the Calvin cycle, and that's what we'll look at next, okay? There's 3 main phases in the Calvin cycle, and you will have time to write this stuff down, so this should definitely end up in your notes. There's 3 main phases, and you will definitely need to know this for exam purposes, okay? The first phase is called carbon fixation, and you don't need to write down this part yet. You'll see it, but you can write it down if you want to. And in that carbon fixation stage, carbon dioxide is brought in. Remember, that is a small non-polar molecule, so it just diffuses in. Your cells are full of carbon dioxide right now, so are mine. It's just diffusing in and out like crazy. Nothing stops it. So carbon dioxide just diffuses right into photosynthesizing cells and then diffuses right into chloroplasts. And some of that CO2 is used to react with a molecule called RUBP, ribulose bisphosphate. You don't probably need to know this full name. RUBP will be handy. This is stuff that I'm going to expect you to be able to interpret a diagram like this on my exams. The AP exam claims to be a little bit more general, but I don't trust them, and we'll come back to that in a little bit. So this is the big piece that you need to know. So carbon dioxide is added to something called RUBP. There's an enzyme that does that work, and we'll talk about it in just a second, called Rubisco. And that is almost certainly going to show up somewhere. So we'll learn about it, but you don't have to write it down yet. The next phase is called reduction. Reduction involves electrons getting transferred to stuff, and that's exactly what happens. The products of this reaction, of the carbon fixation reaction, are transformed just a little bit. They're hardly recognizably different. I wouldn't know the difference between them. I would never be able to tell the difference by looking at them. But they have more electrons. They have energy stored in them. That energy comes from the ATP and NADPH that were made during the light-dependent reaction. So this next phase is called reduction, and it takes energy from ATP and NADPH and puts it into a molecule that is similar to, but not identical to, what we got out of the carbon fixation stage. And then the last phase uses a little bit more ATP to regenerate a ribulose bisphosphate molecule. Okay? So we'll look at each one of these steps in more detail in just a second. This stuff you should have written down now for sure, and then you probably won't be able to keep up with the note taking for as we go through each one of these steps individually. Okay? But I'll try to be really clear about what you're going to be expected to know. Here's the thing. Izzy, you're smiling. That means you're having fun. If you're having fun, you can't possibly be so it's fun to have. So I'm really getting My daughter did that one. Okay, so let's go through these step by step, and then if you have questions you can ask, but like I said, you probably won't have time to get all the notes down, okay? So here's the first step, carbon fixation, and one of the key things that you'll need to know is that the catalyst is an enzyme called Rubisco. And why they want you to know that, I don't know. Whether it's gonna show up on the AP exam, I don't know, but I don't wanna take my chances. If you see Rubisco on the AP exam, I want you to know what it is. It would be really helpful, okay? So here's what happens. We start with carbon dioxide that's just diffused in. That gets added to RUBP. This is what RUBP looks like. You don't need to know this. It's a 5-carbon sugar. That is probably not gonna be super important, but I'm gonna ask you to at least like be able to count these carbons and say like, oh yeah, there's 5 of them, right? So there's a 5-carbon sugar, and that's our RUBP. Rubisco is the enzyme that catalyzes the attachment of another carbon onto that RUBP. The carbon comes from carbon dioxide. This new molecule, we don't care what it's called. I have no idea what it's called. It almost immediately splits, and I don't have any idea how that happens, if it's catalyzed or if it happens on its own, doesn't matter. And that turns into 2 molecules of something called 3-phosphoglycerate, which we'll also call 3PG. And if you're wondering how you're gonna remember all this stuff, the answer is you're gonna do it the same way that I do, which is you're gonna remember it when you need to, and then you're gonna forget it afterward. If you would have asked me a week ago what the names of these molecules were, I would have been able to tell you Rubisco, and that's it. I wouldn't have remembered RUBP. I won't remember, if I'm not looking at it, I won't remember ribulose bisphosphate. I don't remember 3PG, and there's a reason for that. 3PG seems like it should be easy, but then when you see what happens next, you'll be like, oh, you've got to be kidding me. And they are not. So this is the carbon fixation stage. The big pieces you need to know is carbon gets attached to RUBP through the action of an enzyme called Rubisco, and the end result is 3PG, 2 3-carbon sugars, all right? The next stage, so again, if you didn't have time to write all this down, that's okay. The stuff that I told you is what you need to have. You don't need to know the structure of the molecules or anything. And you can get it written down later. The next phase is the reduction phase. This is where we use some energy that we generated in the light-dependent reactions to modify what we just got. What we just got is 3-phosphoglycerate, or 3PG, and you can see this is called 3PGA. Sometimes it's just called PGA. It's a nightmare, but we're gonna call it 3PG. That's all you need to worry about. And then we use some ATP and we use some NADPH, and what we get out of it, you're not gonna believe this, is called glyceraldehyde 3-phosphate, or G3P. So we went from 3PG to G3P. And again, if you're wondering how in the world you're gonna remember this, the answer is you're gonna memorize it right before you need to know it. We'll memorize it right before our exam in here, and we'll memorize it again right before the AP exam. It's not that hard to put in your short-term memory. I wouldn't have remembered this if you would have asked me a week ago. That doesn't stick in my long-term memory. And I will tell you what you'll need to know and what you'll be able to do for the exams. And I know you can do it because I can do it. All right? So the end result of this is we've gone from 3PG to G3P. We have used some energy that we had previously gotten from the light reactions in the form of ATP and NADPH. And that energy has added electrons to our G3P. Hey, come back. There's more energy in this, oh, come on, you can do this. There's more energy in this G3P molecule than there was in our 3PG. It's been reduced, energetic electrons have been added to it. And there's a big, long chemical process, you don't need to know. So at that point, we have G3P, and the next stage is the regeneration stage, and there's a couple of really important things that happen in the regeneration stage one of which you need to know a little bit of details, the other of which you don't need to know like hardly any details at all, okay? So here's the regeneration phase. In the regeneration phase, first of all, one of our G3Ps that are produced ends up just going somewhere else, and something else happens to it. We'll talk about that in a second. And the rest of the G3Ps that get produced end up being turned back into RUBP, that, what was it, ribulose bisphosphate, God bless mine. That requires ATP. So this RUBP is a little bit more energetic than it would have been without the addition of the energy from this ATP.
Wait, the G3P turns into RUBP.
Yeah, I'll go through it in a little bit more detail in just a second, but basically what happens is out of all of what we just did, we end up with what we started with, and we've used a bunch of energy in the form of ATP and NADPH, and this right here is what we really care about. And paradoxically, despite the fact that this is what we really care about, this is the thing that you need to know the least about. Here's what happens with this. This gets turned into glucose. This is a 3-carbon sugar, and that 3-carbon sugar is the basic ingredient in glucose. And you don't need to know the details of this. All you need to know is that little part of the Calvin cycle where a G3P is spit out, that ends up being where all the energy gets stored in the form of glucose, okay? I'm gonna go through this, you don't need to write it down. I'm just gonna show you because for me it's helpful. Come on, you can do this, you can do it. Okay, so the production of glucose happens in the cytoplasm. That G3P gets exported from the chloroplast into the cytoplasm. It's not part of the Calvin cycle, it's not part of photosynthesis itself. Plant cells or producer cells just have that G3P floating around in them. And through a bunch of other reactions, including NADPH and ATP, you end up with glucose. And like I said, that's not part of our required material. So here's our G3P, and here's where you get glucose. That's all you need to know, is that it's used to make glucose. And it's funny that they don't really emphasize that, because that's the whole thing about photosynthesis, is that